He lifted the skull of a loved one. His fingertips met something warm and soft that was not the home of the soul, but a seed planted by death itself. 1957. The rainforests of the South Pacific. The 4 tribe women suddenly burst into piercing laughter. Their bodies convulsing uncontrollably, whispers spread among the tribe. This was Kuru. Soon the victim would lose the ability to swallow and starve to death. An American doctor, Carlton Gideashchenko, noticed a pattern in this mysterious disease. The dead were all women and children. Before long, he uncovered another clue at a funeral ritual known as endocannibalization. In order to keep the spirit alive, women would crack open the skulls of their loved ones and share the brain soft as tofu with children and fellow tribe members. But this was no sacred ritual. It was a feast for death. The 2 clues converged. Gideashchek concluded the secret lay in the brain. He immediately sent infected brain samples back to the lab. What appeared under the microscope chilled everyone to the bone. No living pathogen could be found, yet the brain tissue was riddled with holes. This image caught the attention of a veterinarian, William Hadlow. He exclaimed that it looked exactly like the brains of mad sheep on British farms. Sheep with scrappy would frantically rub against fences, gnaw at their own flesh, until they were bloody and raw in 1968. Gideashchek injected brain material from Kuru patients into chimpanzees. After 18 months of eerie silence, the chimpanzees began to tremble. As if by fate, the experiment confirmed it. Kuru-infected brain tissue was contagious and had a long incubation period for his work. Gideashchek won the Nobel Prize, yet until his death, he believed the culprit was some kind of slow virus. Then came 1972. Stanley Prisoner, a researcher in neurology and virology, treated a patient whose brain showed the same sponge-like damage. He began his battle against this mysterious agent, trying to identify its nucleic acid. But no matter what tests he ran, the data pointed in a bizarre direction. Destroy the DNA and RNA, the agent remained alive. Break down the proteins, it died. The strangeness lay in this. Biology was built upon an unquestioned doctrine, the central dogma of molecular biology formulated by Watson and Crick, discoverers of the DNA double helix. It declared that nucleic acids were the sole carriers of life's information. In 1982, Prisoner proposed a bold hypothesis and published it in a scientific journal. This pathogen might contain no genetic material at all, but instead be made purely of protein. A prion, it was like declaring in a church that God might not exist. The scientific community erupted. Ridicule and skepticism poured in from all sides while debates raged. A real disaster was quietly unfolding in 1986. On a British farm, a cow named Daisy began behaving strangely. Once docile, she became agitated. Her hind legs trembled as she walked when veterinarians examined her brain. They saw something horrifying: dense tissue, riddled with tiny holes like a sponge eaten hollow. This was bovine spongiform encephalopathy, soon known by a more terrifying name, mad cow disease. Investigations quickly revealed the transmission chain to cut costs. Livestock feed was made from the remains of dead cattle and sheep and fed back to healthy cows. The nightmare escalated on March 20, 1996. The UK Health Secretary admitted in Parliament that the disease might have spread to humans through beef, causing a new illness variant, Creutzfeldt-Jakob disease. For the first time, a prion disease had crossed species. Fear spread like wildfire. The EU banned British beef. Overnight, households searched their refrigerators, wondering if every steak hid an invisible killer. At the root of it all was a microscopic catastrophe of protein misfolding. Now we must zoom in to the molecular level. In every human brain, there are normal prion proteins neatly structured like finely coiled springs, maintaining neural health. But at some moment, one of them may misfold due to mutation or infection, transforming from a spring into a jagged shape. That is a prion. It escapes cellular control like a zombie in a movie. When it encounters a normal protein, it induces it to misfold as well, creating another prion. Though this chain reaction is slower than a nuclear reaction, it can still produce exponential destruction. They stick together, forming hard amyloid plaques and fibrils, which accumulate in the brain. These aggregates compress and block neurons, leading to widespread cell death inside the brain. It is like a silent fire has burned everything down, leaving only charred remains, even after the host is buried or cremated. These stubborn, misfolded structures can persist on meat or medical instruments waiting for the next victim. They have no desire, no consciousness, with nothing but their three-dimensional structure. They carry out a massacre across life itself. Faced with this invisible enemy, humanity did not stand still. Feed bands cut off the transmission chain. High-risk tissues were removed to ensure food safety. Medical instruments were sterilized under extreme conditions. Strong alkali 134 degrees Celsius high-pressure sterilization to block medical transmission. In 1997, prisoner alone received the Nobel Prize. The citation read, "He established an entirely new biological paradigm." This did not overthrow the central dogma. It expanded it, just as relativity did not overturn Newtonian mechanics, but defined its limits. It reminds us nature will always find ways to surpass even our greatest imagination.